Hello everyone, welcome back to Chinmoy's Biology channel. So today we are here with the last and the final part of reproductive health, that is infertility. So today we will be going to discuss about all the methods in which the infertility can be solved and how the infertile couple they can conceive through various important methods. So stay tuned throughout the whole episode and don't skip in between, so that whatever important topics we are going to discuss today, like the in vitro fertilization, embryo transfer, gamete transfer, and also that all topics we will be covering in throughout the full episode of today's topic. So stay tuned throughout the whole episode. Let's get started. So what we are going to study today, we are going to study about the assisted reproductive techniques. That is what? That is, there are many couples who are unable to conceive and they are infertile. So when we are studying about the reproductive health, it is very important for us to discuss on the main methods which will help those couples to be fertile, or rather to conceive and give birth to a healthy baby. So this is also a part of reproductive health. So what all we are discussing today? We will be discussing today about the in vitro fertilization, embryo transfer, test tube baby, zygote intrafallopian transfer, gamete intrafallopian transfer, intracytoplasmic injection, and artificial insemination. So these all are very important. Stay tuned till the end of this lesson, and all will be discussed with diagram, and all will be discussed with individual definitions, and what are the advantages and disadvantages in each case will also be discussed. So let's get started. So here comes the in vitro fertilization. So what does this mean? This is a process of fertilization actually, where the egg is combined with the sperm in vitro. So what does this term in vitro means? That is in glass. This fertilization method is taking place in glass. It is not taking place inside body, and the process involves monitoring and stimulating a woman's ovulatory process. So how this women's ovulatory process is stimulated? It is actually hormones are being injected to stimulate the ovulatory process, and removing an ovum or an ova or eggs from their ovaries is the main thing, and then letting a male sperm fertilize them in a culture medium. So that is happening in a laboratory in a glass dish. After the fertilized zygote egg undergoes embryo culture for 2 to 6 days, it is then transferred by a catheter to the uterus. Okay, so the embryo culture is fully done outside. So the ovum is being taken, the sperm is being taken, and the embryo culture is done on a petri dish or on a glass outside, and then it is transferred by a catheter inside the uterus. So this is how we can tell that it is an intention of establishing a successful pregnancy. So this is in vitro fertilization, and it is of too much use nowadays, and it is also one of the assisted reproductive techniques, right? So it is one of the main assisted reproductive techniques. It is also called IVF. Now let us move into the diagram of this, how it is happening. We'll go through that. So this is in vitro fertilization. So what is happening here? First of all, I told you that ovarian stimulation, that is hormonal therapy, is done. So ovary, by several hormonal injection and all, ovary is stimulated. The egg pickup is done. Okay, so the egg pickup is done from the ovary after ovulation. Then this is a vacuum pump. Through this, the ovum is sucked out through suction method. And we can say that this is a hollow needle which is taking out the ovum. So it is under ultrasound control. What is happening now? The eggs, now we are taking a sperm preparation is done here. Then the ovum and the sperm are together fertilized here. Then egg preparation together with the sperm and the ovum is done. The embryo development is done, and then in the blastocyst stage, with the help of a catheter, it is inserted into the uterus. Okay, so here after being inserted into the uterus, if the uterus of the woman is healthy, and it will be able to carry the baby till 9 months. So the implantation and the process after the implantation will happen inside the women's womb itself. So till then this process is done. Okay, so the preparation outside, the sperm, the ovum, they are together fertilized, and what is happening there, they are developed into embryo, and in the blastocyst stage it is injected inside the body of the women through a catheter, right? So let us move on to the next topic. So after in vitro fertilization, we are here with the test tube baby, right? So test tube baby is actually same as that of in vitro fertilization. The embryo is made, it is actually the embryo is mainly made in the test tube rather than in the fallopian tubes, and the eggs and the sperms are fertilized in the laboratory dish, and this process of fertilization which happens in the dish or petri dish is also called in vitro fertilization. So in our common, if a common man is speaking, that the baby is formed through test tube baby, as the ovum and the sperm, they combine together and the process of fertilization is happening in the test tube itself, so they are telling it test tube baby. But scientifically we will tell that the in vitro fertilization has happened. So same thing is happening, the embryo, and that is the eggs and the sperms, are fertilized in a laboratory dish, and this process of fertilization is happening in glass or a petri dish and is called in vitro fertilization, right? So let us move on to the next one. Now comes this embryo transfer. So what is happening there? It refers to a step in the process of assisted reproduction in which the embryos are placed into the uterus of the female with the intent to establish a pregnancy. So the intention is same as it was in vitro fertilization, as it was in test tube baby. Same technique. This technique, which is often used in connection with in vitro fertilization, may be used in humans or in other animals, in which situations and goals may vary, right? So embryo transplant can be done at a day or a day 2 or 3 or later in the blastocyst stage, which is first performed in 1984. It was first performed in 1984. Factors that can affect the success of embryo transfer include the endometrial receptivity, embryo quality, and embryo transfer technique. So the embryo transplant technique should be very much effective. So, and the endometrium in the uterus lining, that is the endometrial lining, should be receptive enough to receive the embryo so that the implantation happens properly. The quality of the embryo should be good so that the next processes, the implantation and the pregnancy and the full term of pregnancy should happen in a proper manner. So this is embryo transfer. So what is the technique? Let us see the diagrammatic representation. So here you can see the egg retrieval is happening. This fertilization process is done. Embryo monitoring and development is being done. Now in this embryo, cryopreservation is done, and it is still this blastocyst stage. It is preserved. It is we can say that it is being processed till it is reaching the final blastocyst stage. Then the blastocyst is inserted or injected into the uterus. Okay, so embryo transfer is done into the uterus, and here again the implantation will happen, and it will continue with the 9 months of pregnancy. Okay, so this is the embryo transfer process. So see, you can just relate this. This is similar to in vitro fertilization, similar to test tube baby. So here and there you will see so many infertility clinics are there. So what they all are doing, these infertility clinics they are taking the specimen from the sperm from the men and the ovum from the women, and they are fertilizing it in the laboratory, and then they are inserting it again to the uterus or the womb of the female, so that the pregnancy, the normal pregnancy, can happen for 9 months. So the initial procedure is like this. So let us move on to the next procedure. Now, the next technique is zygote intrafallopian transfer. So, zygote intrafallopian transfer is an assisted reproductive procedure, and it is similar to in vitro fertilization and embryo transfer only. So, what is the difference? The difference is the name here. You can see it is the intrafallopian transfer. That is, it is the fertilized embryo is transferred into the fallopian tube and instead of the uterus. So, it is inserted in the fallopian tube instead of the uterus because the fertilized egg is transferred directly into the tubes, and the procedure is also referred to as tubal embryo transfer. So instead of the uterus, what we were seeing in the in vitro fertilization as well as in the embryo transfer technique, in this technique they are transferred to the fallopian tube. From fallopian tube only they come to the uterus after covering implantation, all that blastocyst and all. So that process will happen automatically after they are inserted into the fallopian tube. This procedure is more successful than gamete intrafallopian transfer. This gamete intrafallopian transfer we will be studying next, because your physician has a greater chance of ensuring that the egg is fertilized. So it is in this case, the egg is fertilized, then you are inserting it into the fallopian tube. But in the gamete fallopian transfer, that is, the sperm and the ovum are individually injected in the fallopian tube. Okay, so that process is different, a bit different, and here already you are fertilizing the egg and you are putting the fertilized egg inside the fallopian tube. Okay, the woman must have a healthy uterus as well as the tube should be healthy, so that this GIFT has to work. So who all are undergoing this treatment? Might be they are having some problem in ovulation, or the ovum is not being produced, or from the ovary they are not reaching the fallopian tube, some or the other problem if it is there. So GIFT you can do for this. The advantage of GIFT is that it's clear fertilization has taken place, and with GIFT there's no way to know, right, the egg and the sperm are combining to make a zygote, that is an early embryo or not. So here already the zygote is being transferred to fallopian tube, and in the gamete intrafallopian transfer we are transferring the sperm and the ovum. So it is the gamete. So the fertilized zygote is not transferred there. So that mere difference is there. So there are chances of success in pregnancy in this GIFT. Okay, is it clear to all of you? So many different techniques you are learning today. Just to remember what are those techniques, what are the important assisted reproductive methods, and it is a very important part of your reproductive health lesson. So please go through it. Now next, let us move on to this diagram. So this is what is happening. See, you can see the same thing is happening. The ovary is being taken out, then the ovary and the sperm, the semen is here. So they are fertilized. Fertilization process is happening. Now the fertilized zygote is being transferred into the fallopian tube here. So the same technique, you need to just know where it is inserted into the uterus and in which it is inserted into the fallopian tube, right? Now here is gamete intrafallopian transfer. I have told you in the previous slide only when we were discussing about the zygote intrafallopian transfer. So here the gamete is being transferred into the fallopian tube, a tool assisted reproductive technology against infertility. Eggs are removed from the woman's ovaries and placed in one of the fallopian tubes along with the man's sperm. The technique was first attempted by Steptoe and Edwards, and later it was pioneered by an endocrinologist, Ricardo Ash. So it allows the fertilization to take part inside the women's uterus. So the fertilization process is taking directly in the uterus, and just only the sperm as well as the ovum is injected into the fallopian tube. Now with the advances in IVF, the GIFT procedure is used less in pregnancy rates. In IVF they tend to be equal or better, and they do not require laparoscopy when the egg is put back in. So in here it requires laparoscopy. It is a method of surgery, right? So in other methods like in vitro fertilization, they don't require the method of laparoscopy. So that is more successful than gamete intrafallopian transfer. So this was a bit older procedure. Now in vitro fertilization has come and it is mostly used, and GIFT is also used, right? Okay, so let us move on to the diagram here. So here you can see the eggs and the sperm, they all are injected inside the fallopian tube so that the fertilization can take place here, and then full term 9 months of pregnancy will be happening in the uterus itself. Now here next comes the intracytoplasmic sperm injection. So as the name suggests, cytoplasmic sperm injection. So what does this mean? This is a procedure in which a single sperm cell is injected directly into the cytoplasm of an egg. So a single sperm cell, it is a very difficult process. Out of so many sperm cells, you are taking one single sperm cell with the help of a needle and you are injecting it into the cytoplasm of the egg. Now this technique is used in order to prepare the gametes for the obtention of embryos, and they may be transferred into the maternal uterus. So after this injection is happening, then fertilization or zygote formation will happen, and then that embryos will be transferred to the maternal uterus. And with this method, the acrosome reaction, in this method the acrosome reaction is skipped, because acrosome reaction is the reaction where the sperm with the help of its tip, several hormones are being released there, and now there what is happening, the acrosome reaction we are skipping, and generally we know that the tip of the sperm, they are producing several hormones which are reacting with the surface of the egg, and the surface of the egg is then allowing the sperm to enter. So in this case no such thing is happening. With the help of an injection, we are just inserting the sperm inside the cytoplasm of the egg. So we are skipping that process. So let us see what is the diagram showing. See, you can see here inside this hollow needle, we are having a single sperm which is being inserted into the egg cell. Okay, the egg cell is in the petri dish, and the sperm within the hollow needle, with the help of this, it is inserted inside the egg. So this is the intracytoplasmic sperm injection. So this is what is happening here. Okay. So what is this artificial insemination? It is the deliberate introduction of the sperm into the female cervix or the uterine cavity for the purpose of achieving pregnancy through the in vivo fertilization. So by means, we can say that other than sexual intercourse, this is a fertility treatment for humans in common practice, and animal breeding also it is done. Okay, so this artificial insemination may employ, we can say that these techniques, they are actually, the sperm is already inserted into the cervix of the female, because in this cases, generally if the sperm quality or the production of sperm is not good, then in this we can say that without sexual intercourse also, with this process also, we can achieve this pregnancy method. So this is also done in animal husbandry, as well as it is also achieved in certain, we can say that in certain frozen bovine semen specimens are also there. In pigs also it is happening. So this system is also used in animal husbandry as well. Okay, artificial insemination technique available includes intracervical insemination and intrauterine insemination. Okay, where the gametes from a third party are used, and the procedure may be known as assisted insemination. Okay, so the semen or the introduction of the sperm inside the cervix of the uterine cavity for the purpose of achieving pregnancy through in vivo fertilization. So the fertilization will happen inside the body. It is not happening outside the body or in a glass petri dish or in a test tube. So only the sperm is being injected, or the semen is being injected inside the female body. So this is what is happening in artificial insemination. So how it is happening? So this is only the process of artificial insemination. Only the sperm is being injected in the cervix of the female body, and this is the last method of assisted reproductive techniques. These are all the methods through which we can combat infertility, and there are success rates in most of the cases. IVF and all, or we have achieved so many successful babies through this IVF procedures. Artificial insemination and all are also being used in human as well as in animal husbandry. So these are all important important techniques. You should go through all the techniques. Don't skip my video, and more important lessons are going to come. The next lesson which I'll be teaching it will be on the genetics, heredity and all. So you have already studied in the previous classes, but now in 12th standard you will be studying in details about genetics, monohybrid, dihybrid, cross, about laws of Mendel and all. So genetics and evolution is the next lesson, and we will do it in details. And stay tuned and subscribe to my channel so that you can keep on getting the updates what I'll be teaching next. Go through the previous 2 episodes of reproductive health as well, so that it will be easier for you to understand this part. Stay tuned, stay updated, share it with all your friends. Thank you, and always keep watching my videos.